So the speaker today is Patriarca. He's been with us for a while already, so all of you know him. He's going to talk about noise and diversity effects in a homeostatic model for the wake-sleep cycle.
So this talk is about the wake-sleep cycle. Maybe I should say a few introductory words before beginning the real talk, because the topic is very general. Actually, first of all, let me say that this is a collaboration within the BioSim network, and I've collaborated with Emilio and Braun here, and with this researcher, Postnova and Braun in Marburg University. And a few words, because the sleep is quite a large topic, and I'm going to talk about something very particular. So there are many things connected to sleep. And one thing is, for instance, that it's a crucial topic, because, for instance, sleep, you define sleep when you compare it with wakefulness, and then if you are really looking for understanding of sleep, you have to understand wakefulness and then mental activity. So it's a central topic. This is not what I'm going to talk about. We are a long way from modeling these things. All we can model is some numbers and some phenomenology, which is already very complicated. And we cannot even try to model other problems like the REM sleep. The REM sleep is another problem because, as you probably know, it's more similar to mental activity rather than to normal sleep. And the origin of this REM sleep are at the moment an open problem. So what are we going to talk about is just a small part of this general problem, and it's about the wake-sleep cycle. So just the numbers, just the phenomenology, how to make a model which reproduces some features of the wake-sleep cycle. So for this, in particular, we are going to make a model and try to see the influence of disorder and heterogeneity of neurons on the response to an external stimulus, and which is a circadian stimulus and or external stimulus, as I will explain. And the reason is that we make a microscopic model of wake-sleep cycle with neurons, with neuron models, while so far there are only macroscopic models based on linear dynamics of this phenomenology. And I will say something about the Orexin, which is or called also Hypocretin, which is a neuropeptide of central importance for this model. And then I will explain the starting point, which is a two-neuron model. So a model made only of two neurons is still quite good to predict wake-sleep cycle, despite surprising. And we are going to generalize it because we want to study diversity and heterogeneity. So first of all, we know that the sleep-wake cycle is generated endogenously, which means inside in the inner mechanism of the brain. And we know that there is at least one part which is due to a circadian process, so a part of the brain which auto-organizes and like a clock and gives a rhythm. But there is also a homeostatic process which tries to keep the brain working on a constant level of activity. The circadian clock is already a big problem, and there are many models about this. The homeostatic process is still quite not very studied. So you know that the circadian process has more or less a 24-hour split, and basically results in as a pacemaker. And we don't study this, so we take this as an external parameter, an external clock coupled to the system. Instead, we focus on the homeostatic process. The homeostatic process, now it's known that it's mainly due to, or at least there is one important component, which is this Orexin neuropeptide, produced in some neurons in the lateral hypothalamus. And the point is that we want to see how to model this, and we show that if neurons are diversified, the homeostatic role of orexin, so orexin as a homeostatic regulator, is much more effective and results in a much more elastic and robust process. So lateral hypothalamus, I don't know if you can imagine where it is somewhere. It's a small area, and even smaller the one containing the orexin neurons, which are overall a few thousands. Found out in 1998, and they were found to have projection basically to all areas of the brain. It's a process, it's a type of neuron which you can find in many species because it's quite old. Not so old, but quite old. So all superior animals have it. Why is it so important? Originally it was thought to be related to appetite and metabolism. And then a lot of surprises came. For instance, now we know that it's related to wakefulness. If you don't have orexin, you just fall asleep. It's very important for neuroendocrine regulation, cardiovascular functions, and stress conditions. For instance, adding orexin to an animal produces a behavior of feeding, locomotor activity, and chewing, even chewing non-eatable things, which is known to be related to stress condition. And then it's known that this type of regulators are responsible for many diseases. Actually, coupled circadian clocks can produce a lot of diseases. And it's now shown to be also related to addictive behavior and neurological diseases. So it's a very central quantity, a very central neuropeptide, which is responsible of many important processes. One in particular is of interest here, and it is narcolepsy, because this is a sleep disease. Narcolepsy is quite spread, and it consists in inability to maintain wakefulness. So an individual who has narcolepsy just fall asleep, and during these sleeps there is REM sleep, which is unusual because to reach REM sleep you have to fall asleep and then wait half an hour or an hour, something like this. But these REM sleeps are very unusual. They last a few seconds and they are accompanied by cataplexy, so the muscles are totally relaxed, like in normal REM sleep. And they can come and go very fast. And after many experiments, we know that narcolepsy is due to a lack of orexin. And this can be due to two reasons. One is the neurons are absent. There are no neurons producing orexin. The other one is that the receptors don't work well. So the orexin is transmitted from these nucleus in the hypothalamus and received in other neurons. And if this don't work, it's basically the same effect. So this is our K quantity that we want to model. Just to show you an example, this is an experiment on mice taken from this reference, and they genetically prevented the mice from producing orexin. There are two orexin 1 and 2, called or A and B. And so these are normal mice for comparison. And this is sleep. So you see when there is a pulse, it's sleep. And you see that the REM sleep, which is the higher red point, comes only after the normal sleep, which is normal behavior. If orexin 1 is missing, you can see that it's much more irregular and there are many REM events. If orexin 2 is missing, also it's very irregular, but if both of them are missing, and this happens where these orexin neurons are absent or they don't work, you have a very confused situation with narcolepsy. This is exactly what you see in narcolepsy. This is called hypnogram, and represents sleep and wake. And you see that the REM sleep is sudden and with no previous normalcy. So what do we know about orexin? Okay, we know that it causes narcolepsy, and we know that it's modulated periodically by the circadian clock, so it must be coupled somehow to the circadian clock. At the same time, it is produces and increases and decreases independently of what happens. So this is the homeostatic regulation. So there is some mechanism which consumes and produces this orexin. And there is a positive feedback with glutamate, which is exchanged with the other neurons in the brain. And it has been proved that there is a positive feedback by coupling these connections. Another important thing is that this neuron works just below the threshold. They are always ready to become active, and so they work really under threshold. And so what we want to model is these things, considering that when one is sleeping, orexin producing neurons are silent. When we are thinking, we are working, they are firing, like in normal mental activity, but they are not, they are only work is to produce orexin. They don't take part to mental activity, but they are essential because they produce this orexin. And so this is our starting model, a two-neuron model. One represents the orexin producing neuron, the other one represents, say, neurons in the prefrontal cortex, so it could model a mental activity. They are representative neurons in the sense that we just want to see what happens when they interact. This is the model in this reference. So there is the model is like this. The circadian clock is coupled to orexin producing neurons because we know the production is coupled to the circadian clock. Then they project orexin to these neurons, to the prefrontal cortex, for instance, and we know that there is a positive feedback with glutamate. So both of them project to each other glutamate.
Marco, I'm
yes.
Because orexin is a receptor. Glutamate is a neurotransmitter. Those are different things.
Sorry, can you say it again?
Glutamate is a neurotransmitter. Orexin is a receptor. So neurotransmitters are in presynaptic part, receptors are in postsynaptic. So I'm a little lost in the model now.
Let's say that they have very different underlying mechanism of transmission. Yes, exactly. But in the model, I will show you we use the same Hypocretin-like model, and the difference between these processes is just in the time scale, because orexin is related by G proteins receptors, and the glutamate is very different. For simplicity, so we take a microscopic model, but not so microscopic. So we just use Hypocretin and we put different time scales. And the main difference I will explain is that orexin has a very slow time scale, which is the time scale with which it's produced here, and glutamate exists on no time scale. But for the rest, in this model, there is no difference. No, we don't look at the microscopic mechanism. Okay, so that is the model in pictures, and now we try to put the model in equations. Actually, here still a more detailed picture. So we are going to model this as huge Hypocretin neurons. So they have their own dynamics with their currents, and I will show you in a second. But in more, additionally, they have these couplings, and there is a circadian clock acting on this neuron. So I call A the neuron producing orexin, B the neuron which we could imagine responsible for activity or responses of any kind. And now I will go through some technical details, but very fast. I just want to tell you the various terms very fast what they are. So the first three terms here are the normal Hypocretin dynamics. Actually, this is not exactly Hypocretin, it's a little simplified model. And they are the leakage current, sodium, and potassium. Then this glutamate and this orexin are the coupling between the two neurons. I will tell you how in a moment. This is the circadian clock acting on a neuron, and these are noises that we are, and we are going to study the effect of these noises. All the currents are modeled in this way, like in Nash-Nash model, but with a difference for these currents. For instance, this is the leakage current, is just a damping pair. This relaxes the potential, tries to relax the potential toward this value. And these analogously are also currents, but there is an activation variable which tells you if this current is active or not. It can be 0, so there is no current, or it can be 1, near 1, and then this current is working. This depends on the potential of the neuron. So for instance, this current of neuron A depends on the value of VA. And if VA is very small, it's 1. If it's very large, it's 0. So this works as an excitable system. You may already know this. And when the applied signal is strong enough, there are cycles induced. The same thing happens for the potassium. There is the damping pair and there is the activation variable. And in this case, there is a time constant associated to this. It's a slower constant because it's bigger, so we cannot just put it as a function of the potential. And this, so this is the equation as it is. So there is these equations, and these are regulated in time by this equation. And this is exactly the same state function. I go very fast because I don't want to stop on this so much. Now, glutamate. The glutamate is different because even if the form of the current is the same, this activation variable is coupled to VB. So this depends on VB, and B depends on A. So this is the coupling. And analogously for the Orexin. So this is the Orexin current, and I was mentioning it's exactly the same form, but with different time scales and different values. And this is how the activation variable of Orexin is evolving in time. I don't tell you really many details of this, but I just tell you that it's very similar to the previous one, but depends also, there is also another variable which you can imagine as the concentration of Orexin. So this is somehow the central quantity. The only thing to know is that this quantity is 0 or almost 1 when the neuron is spiking. So when the neuron is spiking, the Orexin is consumed very fast. This is when one is thinking, is working. When one is sleeping, then the neuron is silent. This quantity is 0, and then this variable recovers its maximum value, which is 1, with a time constant tau plus. The Orexin grows, then B grows, and this current grows, and then there is the signal which makes the neuron B to spike again. So technically it seems maybe a long story, but it's very simple because it's a simple model. What is the circadian clock? Well, this is not a circadian clock actually, but there are simulations also with different forms of the external current. In this case, we wanted to simulate a stimulus at the beginning of the day. So suppose tau is the period of one day, and at the beginning of the day you have, I don't know, the alarm clock playing or somebody calling you, and then this should be enough to wake up an individual and keep him in wake for a long time. So the external current is just a signal at the beginning of the day, strong enough maybe, but just a signal very short. So if you model the day long 24, actually this is second because we have to take a reduced time scale, but it mimics a day, then this signal is only 0.5, and the rest of the day is 23.5. So there is this periodic stimulus appearing at the beginning of each day. And finally, Xi-A and Xi-B are noises that mimic the noise, the current noise present in neurons, with effective temperatures, or let's say with disorder intensities, DA and GP. So what happens, and how does the model work? So these are the impulses I showed you before. and you have one each period. Now the parameters of this stimulation are such that the system is a little bit under threshold, so it doesn't work regularly as we want. So the individual should wake up each period. Instead it wakes up one day and then the next day sleep. Then this day is waking up and this day sleep. And more or less it's okay because you see that the waking lasts 2/3 of the day, more or less. And this is the p-neuron, so the one representing mental activity. And this is the A neuron representing orexin production. This is the activation variable for orexin, and this is the K quantity representing orexin availability. And you can see that when the neuron A and B are spiking together, and I didn't show the other neurons and the glutamate degrees of freedom, but they are active as well, orexin is consumed very fast. Then, during this day of sleep, you can see that orexin is produced again until the maximum value. And you can see from here that the order of magnitude of this process is 1 day, more or less. And then it's consumed again, and then again. Why doesn't it start here? Because it's too low. So this is how the homeostatic model works. If the orexin accumulated so far is not high enough, it will not wake up, it will sleep the whole day. Here, of course, it's very high, so it's enough to wake up the individual, and then it will be woken up for a big part of the day, and then it will fall asleep again at some point during the day. So this is an example, and this is the enlarged part. This is a blow-up of this part where there is no spiking, and of this part where there is a spiking. So you can see more in detail as it works, and you see the difference in the initial level of orexin and how the details work. You see that the orexin is produced, so this begins to spike and tries to wake up these neurons. Here it doesn't manage, and here it manages because there is enough orexin, so the current is big enough. But this is just an example. What we really want is something like this. We want that each day the neuron wakes up for, say, 16 hours, and then there is 8 hours sleep. So this is nice, but it's made with an optimal tuning. Yes?
But the model you show, you sleep for a whole day.
Sorry?
In the model you show,
Here,
you sleep for a whole day.
Yeah, this is because we intentionally have chosen this parameter. I don't know if it was I hope I've not confused you. So this is what we want.
Yes.
Putting optimal tuning.
Yes.
And this is just an example to compare what happens when one doesn't manage to wake up in the model.
Okay, so the model also gives you a period 2 solution where you sleep for a whole day and wake up only every other day.
Yes, if you change some parameters, for instance, you put a larger impulse, initial impulse, you can make the individual wake up every day. So this is an intentional situation to show that it may not work depending on the parameters. And this is exactly the point, because, yes?
So, about the parameters, how many parameters do you have?
There are a lot of parameters. The point is that the microscopic parameters of the Hutchinson models are more or less fixed, because we take them from the literature. What is not known is the glutamate and the orexin parameters are some educated guess, but they are well justified. The time scale should be more or less correct, because we want the day time scale, and because the glutamate time scale also is known. But to answer precisely to your question, let's say that each current has a conductivity, an equilibrium potential, and then if you put a sigmoid function for the response of any activation variable, this is W is one more parameter, and here there is one more parameter. But these parameters are more or less known, so we don't invent them. So they are taken from the. But still, what I'm showing to you is quite. You can vary this parameter a little bit, and the results are more or less the same. There is some uncertainty in this parameter, but you cannot vary them too much. So let's say that we cannot claim that this is representing the actual wake-sleep cycle, but that it is a realistic model as long as the time scale are conserved. And anyway, the point here is more to show the effect of diversity. So, in this stage, the realistic, really precise value of the parameters is not the main issue. We take realistic parameters, but not really, we don't have precise experimental data to fit apart general wake-sleep cycle. So yes, I was saying that this is the key point that we were mentioning, because if you tune the parameters, you have a good sleep-wake cycle. If you tune them in another way, you have a bad wake-sleep cycle, like with double periodicity, and you don't want this, of course. Now, if you make a generalized model, you can have a much more resilient model, which is more elastic, and you have a whole parameter range where you have a good wake-sleep cycle. And this is the point of this work. So it took a long way to arrive here. And now we study one version of a generalized model, in which there are many orexin-producing neurons, and still only one neuron representing T or whatever. And the reason is that the orexin-producing neurons have more heterogeneity than other types of neuron, and it's not known why. So an idea would be that in this way, the response of the system is better. And so it's a natural thing to look, to try to understand what are the consequences of adding some diversity in these neurons. Before doing this, let me show you what happens when you just put noise, either in the neuron B or in the neurons A, each independent noises, with temperature TA or TP. And here we have simply a stochastic resonance, simply in the sense that this is a well-known phenomenon and already found in real as well as model neurons. And qualitatively, this is what happens when you have t equal to 0. The parameters have been tuned in such a way that you have no response. Okay, so this is the pulses this is the pulse actually arbitrary with different periods, but still the point is that when you apply an impulse, there is no response with these parameters. When you increase the temperature in neuron B, you have some spikes. When you increase even more, you have more spikes during the impulse, and so it's reacting. But if you put too high temperature, it's always spiking, and this is something you don't want. Yes?
What do you mean by temperature of the neuron?
Well, forgive me, this it's not temperature, it's a fluctuation intensity. It's the current noise.
Yeah, sure. Initial relation is not.
No, it's just a measure of the fluctuations of the current.
But is that the amplitude of the noise, or is the effect of temperature?
No, it's the amplitude of the noise in the input signal. So in the input signal, you tune the amplitude. In this slide, I kept T, but it's not a good choice. You're right. In fact, in the next slides, I will put D. So D and T are the same quantity. It's just the amplitude. And here I don't show you anything else because it's quite clear qualitatively what happens. Here you have a response which is to rate, and here you don't have any response at all. So there is an optimal intensity of fluctuations by which the response is best. Let's go from the potential versus time to the raster plot. So we put a dot for each spike so that we can represent more data altogether. And we redo the plots for several, okay, here is the D I was mentioning. We redo the raster plots for several amplitudes of the fluctuations. So here again, when there is no noise, 0, I have chosen this is just an example with parameters so that you have double periodicity. It's like before in the examples I've shown. So it's one normal day and one day that one sleeps the whole day, one normal day, and so on. This is double periodicity. This is bad, of course. We don't want this. If you increase the temperature in the neuron, the fluctuations in the neuron B, then you see that there is a gradual improvement until more or less, but it's not so exactly. You have a situation in which every day there is, almost every day, there is a period of wakefulness. But if you increase temperature or fluctuations too much, then there is no real sleep. There are a lot of frequent episodes of sleep and wake, and this is also bad. So, and the same thing happens if, okay, so this is an error. This is it's correct, this is the A. And this is when instead you put the fluctuations on all the neurons A. And you start from the same state, which has double periodicity. But this is a little different. This response is better because you see that almost every day you have a wakefulness period, but it's more irregular later. And this is something we are still trying to understand well.
Here you have many neurons A,
yes,
and they have all noise,
yes,
independent noise, and they are the same neuron, all that identical, or they are also diverse?
They are still identical.
They are still identical.
Yes. So it's just neurons. It's just but you can show that in this case, with the model that we use, it's like having two neurons. So we have checked that two neurons or n neurons which are homogeneous give the same result. The only difference, of course, is that the noise is rescaled in some way depending on the number of neurons. Okay, this as long as stochastic resonance is concerned. So, and now we go to the actual heterogeneous model. So we have our neurons, A neurons producing orexin here, and we have the single B neuron here. And now we really put some difference here so that they are different from each other. And I show you one example of a difference in one coupling between A and B. You can put, actually, we have several couplings, so you can put different diversities in here. So this I mentioned, and so let me just mention in the equations, just a second, where the diversity enters. As an example, I show the glutamate coupling, because the glutamate is coupling the A to B and the B to A. So when we diversify the A neurons, this current, which is this one in the tail, has this activation variable which depends on some threshold. You remember this step function. This is what tells the ion channels when to open and to close. So W is the threshold at which the neuron begins to spike. Instead of putting the same threshold for all the neurons, we put different thresholds. When we do the same thing for the other coupling from B to A, then also in the current B of glutamate, there is this activation variable, and this, since it's only one neuron, we take the average of all the signal. But the point, this is just the technical side, the point is that now the neurons, actually the coupling, have different thresholds. So they are excited, they begin to work at different values of potential. Someone will need a small potential to begin to be active, so a current different from zero, and someone will need a larger potential. And we see what happens now when we do this. So we have these different thresholds for the different neurons. And how do? We keep the same average value so that we can compare with the two-neuron model. We introduce a distribution and we distribute the other values according to the distribution. Same average value, but different variance or standard deviations. And we see what happens. And this is what happens. And it's similar to stochastic resonance, but qualitatively much better, because you can see that this is regular. If you remember how it was in the case of stochastic resonance, it was very irregular. So consider this. This is when the neurons are homogeneous, and they are chosen, the parameters are chosen so that you have no response. It's under threshold. Then you increase the diversity, and you have a better response. You increase the diversity even more, so they have different thresholds. And if you increase it too much, they respond always, so this is very bad. So there is an intermediate value of diversity, which is this one, where you have an efficiency, a good efficiency for the response to an external signal. This is one example of signal, but I want to show you another example which corresponds to the peaks I was saying. So these are the peaks corresponding to a signal in the morning, like the alarm clock. So you have this peak, and then you see how the system reacts. And now we start from another set of parameters like the same one I used before. So with double periodicity. Here the individual wakes up, and here it doesn't, and so on. And this is with zero diversity, which means all the neurons are the same. Then we increase the diversity of the A-B coupling, a little bit and nothing changes. When we increase it enough, then we have a very good response. The response is qualitatively good, so it remains with single periodicity, but the period of wakefulness decreases slightly. Of course, if you increase even more, the system gets even worse. But you can see that there is of course, these are chosen intentionally in this way, and to show better that there is an improvement in the response when we add some diversity. This is when the diversity is added in the glutamate coupling but going from in the other direction, and we have a similar response. So double periodicity turns into single periodicity, and then there is a slight modulation of the signal. But you have a higher wakefulness period for some intermediate value of diversity. And well, we are still trying to understand what is the best way to characterize quantitatively the quality of sleep. For the moment, we evaluate, we divide the day into two parts. The 2/3, when one has to be woken up in the wakefulness state, and the last third, when one should sleep. So the wakefulness in the period of wakefulness in the wakefulness period is counted as plus, and if the individual sleep is counted negatively. And the opposite thing for the sleep period. So we get a coefficient which is something like this. So this is 1 when if one sleeps 2/3 of the times, the first 2/3, and sleep the remaining third, and it's 0 when one does the opposite. So it's constructed in such a way that it varies between 0 and 1. And maybe this is more helpful to see that there is actually a resonance for some intermediate value of diversity. So the point is that what is known as a phenomenological fact about these orexin producing neurons, that they are very heterogeneous, has immediate consequences for a more robust response or the existence of an optimal degree of diversity. The point of the parameters that was raised is good because, of course, to prove that the heterogeneity of the actual orexin producing neurons is the one which optimizes the response is difficult because then we should really know all the parameters and compare it with the result. But in this is a model, so let's say this is just a model system, and in this model system, made with this Huxley model of neuron, it's an immediate consequence that if you have heterogeneous production of orexin, automatically you have the existence of an optimal threshold of diversity. So you, let's say, you should put diversity into these neurons to have a better response. Okay, so I think this is more or less what I wanted to say. And well, this model has dependence on diversity, and especially it shows that there is an optimal amount of diversity, and we can consider this a collective effect. And there are, of course, many other things to check and other parameters to vary to explore the model, which is only explored in half. So I think this is all.
Okay. Thank you. Question?
You choose the parameter values for which the model with two neurons here is period 2. No? So then you have when they sleep, when they wake. And in that, you get that the diversity produces the regular cycle. But if you start with the parameters for the model with two neurons where you already have a periodicity 1, then do you get a better also regularity or whatever by having many neurons? Or
depends, yes. Depends on the parameters where you start. If you start with just above the threshold, you still have an improvement, and then you have a decay. If you start with very high response, probably you don't have this dip in the response. But we know that these neurons work under threshold. So we know that if there is no external stimulus, the neurons are silent. And we know that a small stimulus is enough to excite them. So one of the very few things we know is that they were just under pressure. So this is the reason why we chosen this starting point. And there is actually a much richer behavior because you can have triple, quadruple periodicity depending on the parameters. And when you increase the diversity, you can go from periodicity 4, 3, 2, and you can have all these different types of behavior.
You said 5 neurons is more or less enough to represent qualitatively some significant changes and some significant improvements.
Yes, actually we have used 50 and 100 in the most of simulations, but it's more or less the same, and the actual number is 6,000-7,000. I mean the number of neurons in the...
Do you have any explanation why nature is choosing so many neurons?
That's a good...
many of them will die, or...
Pardon?
Many of them will die one day?
I think it's also because of just quantity, size of the brain, because they project to many regions of the brain. And to do this, you need a lot of neurons, you need a lot of projection. So this could be one reason. And
what
then
do you call projection? Or
pardon?
When you say projection, it sends some information all over the brain?
Yes, it's called synapse. Yes. And they, these connections go to area of the brain which works for many different things, as I mentioned. I mean, from looking for food and stress and hormones and everything. So maybe these numbers, 7,000, is for this reason, because with few neurons you get the same result.
Circadian clock, actually. I mean, you were just putting that as an external display.
For us, it's an external.
I mean, but in the body, what is it?
But it's actually a group of neurons which synchronize in a robust way and produces a periodic input. Actually, some of us have been working on some of these models. And this is by itself another problem. I mean, it's complicated to make a good model of circadian clock. But basically it's let's say it's a complex system which synchronizes and produces a periodic output. And the most complicated thing that we didn't want to address is that there is an article, not a recent article, talking about how circadian clocks are coupled to each other. So if you really want to model a circadian clock well, you have to consider that it's coupled to other circadian clocks. So a typical thing happening when one has a jet lag is that you resynchronize very well the daylight circadian clock, but not the thermal one. So the temperature goes in one direction and your daytime in another one.
Yeah, exactly. That's what I read, that there are many clocks in the body, and all of them have different periods actually. So you have a temperature one, you have one for sleep awake, you have one for the production of some hormones. And if you cut the light, they can go out of synchrony. But what I also read is that sleep awake it has a given period, which is not so. You're not including that.
No, we are not including. This is admittedly a simple model. We not only we put it as an external parameter, but we consider only one circadian clock, which is the sleep time. So this is a starting indication of the model.
Well, related to this, in fact, you always put an external signal, but if you do not put external signal on the circadian clock, do the two neurons have a period, a natural period? Or do they just stay silent?
Good question. I think if you don't put noise, you choose the parameters in a certain way, they remain silent. If you put enough noise, they can give rise from time to time to this period. But we have not checked what is the critical noise and what happens. This we didn't check. But I see that this is a good question because actually.
Because you said that the sleep cycle of a person has a natural cycle that might not be coinciding with the daylight cycle. But of course, the model, these two neurons, do they have a natural period by itself? Or?
The system, Well, it depends if you manage to get started. Yes, of course, it must have appear because they are coupled oscillators, basically. But the point is that if you don't put enough noise or you don't tune the parameters, it can remain always silent. And this is actually maybe a problem because we know that if you don't have any signal, after you sleep, nothing will wake up anyway. So there must be a way that the system knows that you have slept a lot and you wake up. But this is not in this model, at least not with these parameters. We should maybe try some other tuning.
Okay. So we are lucky to have couple of slides. That's